Cool.
Okay, so hi everybody, my name is Gnin Alyousef, and today I'm going to be presenting on the role of AI in proteomics and cancer. So cancer is responsible for nearly 1 in 4 deaths in Canada, making it the leading cause of death. Approximately 230,000 Canadians are diagnosed with cancer annually, and there are a bunch of risk factors, things like tobacco use, poor diet, obesity, physical inactivity, alcohol consumption, sun exposure, environmental exposure, etc. But when we talk about cancer, it's easy to think of it as cells growing uncontrollably. And that's just the phenotype, like that's the outward behavior. At a molecular level, cancer is a complex systems breakdown. And it's defined as a disease that's dysregulated by proteins and pathways. So in a normal cell, you have DNA that gets transcribed to RNA, and an amino acid is made from it, and from there the amino acid is folded into a protein in its native state, and from here the protein can go on in the cell and do its job and interact with other proteins and create a cascade of events. However, in a mutated cell, or when in this case cancer, the amino acid chain or the polypeptide chain can get mutated, which can cause the protein to get mutated, and from there the fate of the protein can either be degraded, or it could go into the cell, and this is again, like I said, usually what happens in cancer. So it's not just that the gene mutates, it's what happens to the protein that gene encodes. How it's expressed, how it interacts, how it gets modified, degraded, sequestered, etc. So to really understand cancer and to treat it, we need to move beyond DNA sequence and look at proteins in context. And this is where proteomics comes in. Basically, proteomics is the large scale, like basically looking at proteins in a large scale. And it gives us a deep look at what's actually happening inside the cells, how the proteins behave, interact, respond to change. To put this into context, one cell has 42 million proteins. In your bodies, there are about 30 trillion cells. So just to show you guys how many proteins there are in a single cell, let alone your entire body. So this is why we need to be able to see how the proteins work. For example, what oncogenes are turned on, what tumor repressors are lost, what are the signaling pathways that are misfiring that cause one cell to grow uncontrollably and become like a cluster of cells, also known as a tumor. So again, why does proteomics matter? Let's think of a factory. We'll think of DNA as the blueprint, mRNA as your instruction manual, and proteins are the actual machines doing the work. In cancer, some of these machines are duplicated, they're broken, they're turned on at the wrong time, and they're wired into the wrong systems. And there's also a bunch of control systems like checkpoints and feedback loops that are also lost, or they also fail. So cancer isn't one thing going wrong, it's a cascade of failures in a highly interconnected system. And this is why looking at proteins is essential, because it tells you what is actually happening, whereas looking at DNA and genomic data tells you what could happen, or what might happen, or what a cell is wired to do. Let's take, for example, let's take 2 identical tumors, or 2 identical mutations, okay? These tumors can have, they can have identical mutations. However, the proteomic profiles can be very different. One may have an active, there could be a certain pathway that may be active while the other doesn't have an active pathway. And many therapies, just so you guys know, in cancer target proteins and not DNA. So this is why like the expression and the modification and the interactions of proteins in cancer is very, very important. So where does AI come in this? As you guys can see, this right here is a cell with a bunch of proteins. It is a very, very, very like crowded environment. It is very complex to be able to say that this protein interacts with this one and this one and this one. Proteins have an insane amount of like they interact in nonlinear ways, and their profile is massive. That is why we could use AI to help decode proteins at a scale of like decode the data sets that are huge and massive. So AI can help us go from pattern recognition also to biological reasoning. So this clustering and inference is something that humans can do, and we can hypothesize. For example, if we look at a couple of proteins here, that this one interacts with this one, which causes a cascade of events, and humans are able to think about this and then think about, okay, what protein interacted with this to cause this effect? This type of reasoning and generation takes weeks for humans to be able to figure out. Whereas AI, if it looks at a bunch of data sets, look at a bunch of like proteomic data sets, we can say that it would be able to make these inferences way faster than humans can. So a more concrete example is, let's say we have 2 breast cancer cells. And in breast cancer, there's one of the mutations is a HER2 receptor, or the estrogen receptor that gets mutated. So let's say we have 2 breast cancer cells. One shows normal expression of estrogen receptors and the other one doesn't. An AI tool can notice this discrepancy, recognize that the genes of the estrogen receptor targets, and propose that one is absent and or dysfunctional. And it can even go a step further and say the proteins that actually regulate the estrogen receptor are dysfunctional and are a potential cause. This real reasoning that we have here is called biological storytelling. And humans can do this, but right now there is no AI that is able to biologically tell you this happened because of this, this, and this. Does that make sense? Okay, so using AI-driven proteomics for precision oncology, there are so many advancements in AI-powered proteomics, and they're paving the way for developing therapies based on patients' unique tumor biology. Each person is very, very unique. Every cancer is unique, and that's why it's so hard to be able to figure out exactly what's going on in every single person and cure cancer completely. Traditionally, precision oncology has focused on genomic information and identifying mutations. However, proteomics offers a dynamic perspective by revealing real-time protein behaviors, interactions, and modifications within cells. With AI's capability to analyze comprehensive proteomic data, we can uncover critical insights, such as identifying an overactive signaling pathway. So AI can detect when a signaling pathway, when signaling pathways are excessively active due to post-translational modifications, for example, rather than genetic mutations. Or understand therapy resistance, for example, by analyzing protein degradation rates and modifications, AI can pinpoint mechanisms behind tumor resistance to certain treatments. Or we can even use it to assess protein interactions. AI can evaluate how proteins interact with tumor microenvironment, which allows us to look at complex networks that drive cancer progression. So by integrating AI-driven proteomic analysis into clinical practice, we can move beyond the static genetic markers and developing dynamic, personalized treatment strategies to adapt to the evolving nature of cancer. This approach enhances our ability to select the most effective therapies, monitor treatment responses in real time, and adjust interventions as needed, and ultimately improve patient outcomes in oncology. However, what are some pitfalls in this currently? So right now, with the databases that are getting updated, they're getting updated by human research. So for example, I would go into a lab, do my research, put it into the database, and then the AI would be able to analyze this database. So AI can only go as fast as I can go as a researcher. Another thing is right now, AI does still have its hallucinations, which is very hard to be able to almost put humans in the hands of AI if it still has hallucinations, especially when it comes to things like treating cancer. However, with Singularity, there's a possibility that data sets can be updated by AI alone, and AI could do its own, find its own data sets without depending on humans to do so, and hopefully the hallucinations will also be gone by then. Thank you so much. Any questions?
So at one point you said that AI can't give you the various steps, but isn't that what chain of thought reasoning is? When you ask an AI model, show me your chain of thought reasoning, it gives you step by step.
Yeah, definitely. I'm sure that like right now with AI, it does do that. And even if the answer is wrong, it gives you an amazing reason as to why its answer is right. However, I'm talking about currently when it comes to data sets that involve proteins.
Right.
AI is unable to use this massive data set to be able to tell us what caused a cancer, and not only if, let's say, one protein is mutated or there's one mutation in a certain area and it caused cancer, it doesn't tell us what caused this mutation or reason its way through to be able to get us to the last outcome of cancer, if that makes sense. Yeah.
So the hallucination problem, the next time you hear somebody talking about this, what you'll often find is every human speaker talking about AI, even if they're not conscious of this, at a subconscious level, they're looking for something that makes humans better than AI, or humans can do something that the AI can't. And so often it's just like what you did, which is to say that if they keep hallucinating forever, you didn't say that, but that's kind of what you meant, then we can't use this. But the amount of, hallucination is coming down, right? And by 2027, when we think that big time artificial general intelligence will exist, the amount of these kinds of errors will be practically zero. So it's important to just keep in mind that as far as we know, there's nothing we can do that AI can't do. I mean, if you don't describe it very well, then AI can't do it until you describe it better, right? But anything that you can clearly describe in words or formulas as a desirable thing to do, AI then will become able to do that better than you can, or better than any human in the long run. Yeah. Other comments, questions? Okay, so Tuesday we'll have 3 students presenting, and so I want you to think about the 8th. That is when we're going to talk about the time capsule. And you guys have more questions than any class has had before. Because as far as I can tell, it's a bit unclear when we should actually start the time capsule. Like, why would we start it in 2025 if Shauna is going to space in 2026, right? Because don't we think Shauna going to space is like a really big deal for this class? Yeah. We know her, and she's going to space, right? Secondly, there's some difference of opinion of where the time capsule should be. So I think it'll be in 2 places. The coroplast cutouts from 2020 and 2021 and stuff will be in my 4-car garage. And then Pram, who is leading the discussion, she's now a first-year medical student at the University of Calgary. She never plans to come back here, so she's totally wedded to Calgary. So I think the main time capsule will be there, even though the course is here. Now you can argue in various ways that the time capsule belongs to the course, but Pram is the person who suggested the time capsule. It was her idea, right? So I think it's perfectly fair. We'll divide it, and part of it will be in Calgary, and kind of small things. Most time capsules have small things. The coroplast cutouts, of course, are life size, right? So they're like 5 to 6 feet tall. So anyway, be thinking of, and So you guys should help us decide, are we still going to start this, to put the time capsule away in 2025? Or should we wait till 2026? And I'm thinking that, you remember when Shauna said she could join us for the social event? I think she was thinking about the 8th. Now, it's going to be on Zoom, that there's no food, there's no party, there's no social event. But still, I think she could possibly come. And also the other interesting thing, Pram wants to write a paper about space medicine, and she's asked Shauna and me to help with it and stuff. So there's a lot to talk about, and I wanted to give you guys the idea that you could actually, if you come up with some bright ideas, change which way it works, whether we start in 2025 or...
Should we put it in space?
Oh yeah, we can put it in space, or put one of them in space. I don't think it would make sense put the core cutouts in space. But on the other hand, isn't there's a Tesla Roadster in space, right? One of Elon Musk's spaceships contained one of his cars, right? Mm-hmm. So that's
we can ask him.
Yeah. So
can we put our time capsule?
That wouldn't be the biggest thing ever. Yeah. Okay. So